Good morning, everybody. First off, I'd like to thank DSA for giving me the opportunity to talk to you today on this topic of signal detection and trending. The talk I've set out to deal with various issues within this. So we will deal with why do we need signal detection, the objectives of signal detection, how we look at our PV databases and how we use these for signal detection. And then we'll talk about what is a signal, how we use data mining to help us in signal detection. Then I'll go through some of the tools we can use to try and identify signals, what we do if we do detect a signal, how do we proceed with it, and then I'm going to go through some case examples I've got of where we've detected signals and why they occurred and how we dealt with them. And then near the end, what I'm going to discuss is how the regulatory authorities are doing their signal detection and some examples of what they come up with. And then finally, we'll have some conclusions to wrap up on the topic. So first off, why do we need signal detection? If you remember, we are very limited in our pre-marketing clinical trials because the trials that we submit are usually of a short duration. So in other words, the effects that develop with chronic use or those that have a long latency period are impossible to detect. And also clinical trials are usually done on narrow populations. So they do not include special groups like, for example, the young, the old to a large degree. And the population is not always representative of the population that may be exposed to the drug after approval. The other thing in our clinical trials is there's usually a narrow set of indications. So in other words, we're only looking at testing for the efficacy which has been studied, and we don't cover the actual evolving use. So in other words, when the product is licensed, when it gets out there, veterinary surgeons may use it slightly differently. Also, the pre-marketing studies we do are of a small size. So in other words, effects that would occur very rarely are very difficult to detect. So in other words, for example, if we looked at the statistics we use for clinical trials, if we wanted to detect an adverse event or adverse reaction which has an incidence of 1 in 10,000 to a power of 0.95, which is what we use in clinical trials, we need at least 30,000 animals in that study to do that. Now, as you can gather, there are very few studies that are set up with that number of animals in it. So therefore, our clinical trials seldom just detect or define all the frequency of all important adverse reactions or adverse events that can occur with a product. And this slide here shows you that actually with the various statistical powers here, but you see that a lot of clinical studies, when they're done, usually you probably only have about 300 animals max in each group. And you can see at a 95% level, you're only going to find a frequency of an adverse reaction, which is there very frequent at 1 in 100. When you get to the 1 in 10,000 here, even if we came across and decided to take a lower level of statistical power, even at 63% statistical power, we still need 10,000 animals in that population to detect the frequency of 1 in 10,000. So therefore, these studies rarely show us the whole safety profile of the product. And then when we do put the product out in practice, there are a larger number of animals that it's used with. Veterinary surgeons will use products in combination with other products. Their environmental conditions are different. We have other species or subspecies. We can have off-label use in dosage or time. And we also have a condition. In other words, we will have the product used in younger animals or older animals or animals that could be suffering from underlying disease. And sometimes we do have product failures. So in essence, the safety profile of a drug evolves over the lifetime of its market, and that's part of what our role is in this job of looking at signal detection. Now, I thought it was interesting to turn the table I showed you on itself and look at it in a different way and look at the number of animals required and the number of AEs detected. So even if we were trying to, with 3 cases, to get a 1 in 10,000 frequency, we would still need 65,000 animals to be treated with that product. So again, it shows you that a lot of the AEs that we're trying to detect in our signal detection process are rare, very rare, so they're much lower than this level here. They could be 1 in 100,000. So that we do have to have a lot of animals treated even to detect those AEs. So that's the reason why we need the signal detection. If we then go on and look at it and say, well, what are our objectives in this signal detection? What are we trying to do? Our first one here is that, as I've said, we're looking to identify the rare adverse events that occur that are not detected in our pre-license studies. We're also trying to monitor increases of known adverse events. So in other words, are they increasing as we put it out into the market? Key one we're trying to do is we're trying to identify risk factors, be that use of other products at the same time, or pre-existing conditions that may lead to these reactions. So in other words, that's the key one we have to look at, is that is there an underlying disease there, or is there concomitant products? And we'll talk about these, some of these in the examples later on, that could lead to these adverse events occurring. And one of our key roles in this signal detection is actually, it's essential to refute false positive reactions, because when we do signal detection, we will get more signals than the false signals than positive ones. And therefore, we have to refute those false positive reactions. So that's also part of the role here, is to see this, to ensure that what might be perceived as an adverse event is a false positive.